It's my pleasure to introduce Ishtiaque Hossain as our next speaker. So Ishtiaque started in science with a degree in biochemistry at McGill University, where he then continued with a PhD in the lab of Dr. William Pastor to study Zik transcription factor in early human development. He had a great track record of presentation and awards, so I'm really looking forward to this talk. Ishtiaque, the floor is yours.
All right, thank you so much for the introduction, Matthew. And I'm also want to thank the organizers for letting me share my work with everyone today. So my project is mostly focused on early human embryonic development, and it really focuses on two stages: the pre-implantation epiblast, as you can see on the left, and the post-implantation epiblast, as you can see on the right. And basically the epigenetic barriers or the drivers that facilitate transition from the pre-implantation epiblast stage, as you can see by the green cells here, to the post-implantation epiblast stage, as you can see by the red cells. And really, we can take knowledge from Waddington's landscape model and say that there are basically a lot of epigenetic barriers and drivers of these processes and these transitions from the naive state to the prime state. So we can model the pre-implantation epiblast using naive HESCs, and we can model the post-implantation epiblast using primed HESCs. And while there have been a lot of studies that look into the drivers and barriers of these transitions, most notably focus on transcription factors that help maintain naive pluripotency or facilitate naive pluripotency, the factors that drive exit from naive pluripotency to a prime state is a little less understudied beyond looking at the pan-pluripotency transcription factors like OCT4, SOX2, and NANOG. So to really focus on this a bit more, we mined ATAC-seq datasets and generated ATAC-seq datasets of our own. And we basically did Motif analysis on loci that are specifically accessible in primed HESCs compared to naive HESCs, which are cultured in two media conditions that we call 5LF and PXGL. And we observed is that among the loci that are specifically accessible in primed HESCs, a lot of these motifs correspond to factors that are known as ZICs. So this is a family of transcription factors consisting of ZICs 1 through 5, of which ZICs 2, 3, and 5 are highly upregulated in primed HESCs compared to naive HESCs. Then we conducted ChIP-seq of ZIC2 and ZIC3, and what we observed quite interestingly is that, and consistent with our ATAC-seq analysis, was that ZIC2 is highly enriched over prime-specific attack peaks, but have virtually no enrichment over naive-specific attack peaks. Next, we also observed that ZIC2 and ZIC3 have very similar DNA occupancy patterns. So a loci that has high enrichment for ZIC2 will also have high enrichment for ZIC3, which will be more relevant later in this talk. Next, we wanted to see what chromatin states were associated with these ZIC-bound loci to get a sense of what functional role they play. So for this, we mined ATAC-seq datasets and also histone ChIP-seq datasets in naive and primed HESCs and cross-referenced that with our ChIP-seq datasets in ZIC2 and ZIC3. What was quite interesting is that the ZIC-bound loci were quite diverse in the chromatin state, and we had 4 distinct clusters upon clustering. Among these, clusters 1 and 2 were generally active in both naive and HESCs. However, cluster 3 corresponded to prime-specific active chromatin, which acquired Ishtiaque acetyl only in the prime state and also became open specifically in primed HESCs, whereas cluster 4, in contrast, although what became slightly more open in primed HESCs, were mostly marked by Ishtiaque-27me3, which is a repressive mark marking poised genes. And so consistent with their chromatin state, when we annotate these ZIC-bound loci to genes, what we observe is that, again, clusters 1 and 2 are very highly expressed in both naive and primed HESCs. However, genes annotated to cluster 3 peaks are very highly upregulated in primed HESCs compared to naive HESCs, whereas genes annotated to cluster 4 peaks are generally kept in a repressed state in primed HESCs. So to study this a bit more in depth and know the functional role of these transcription factors, we tried to generate single and combinatorial mutants of ZICs 2, 3, and 5. However, while we were able to generate successfully single mutants of ZICs 2, 3, and 5 and combinatorial mutants of ZIC2, 5 and ZIC3, 5, we were unable to generate mutants that have deletion of both ZIC2 and ZIC3. And this is highlighted by this, this annexin V staining, which is marker of cell death, where they had a high staining for annexin V in the ZIC2-3 double mutant compared to the single mutants and the wild type control, suggesting that loss of both ZIC2 and ZIC3 results in high cell death. Therefore, to circumvent this issue, we used a transient approach where we use Cas9 RNPs to delete ZIG2 in different ZIG3 or 5 mutational backgrounds, and did our analysis about 5 days post-nucleophector, post-electroporation. And we found quite interestingly is that upon deletion of both ZIG2 and ZIG3, not only do they undergo more cell death, they also undergo more spontaneous differentiation, as shown by CD56, CD184 staining. Next, I'm sorry, just 1 second. Next, we also looked into the RNA-seq we conducted RNA-seq of all our ZIG mutants and wild-type controls. And what we observed quite interestingly is that there were overwhelmingly 2 gene sets. So this is consistent with ZIG2 and ZIG3 having redundant roles, which is why we see minimal loss changes in gene expression in the single mutants. And we also observed there not being any changes in gene expression or very little in the ZIG5 knockout, suggesting that ZIG5 plays a lesser role in the context of prime pluripotency. When we then annotate these genes, so we characterize as gene set 1 and gene set 2, what we observe is that gene set 1 is mostly comprised of genes that are upregulated upon in vitro differentiation to the mesoderm or the endoderm lineages, whereas gene set 2, so these are genes that are downregulated upon loss of ZIG2 and ZIG3, were comprised of genes that are upregulated upon escape from naive pluripotency to a prime-like state. When we then wanted to get an understanding of what was the chromatin state of these loci upon loss of ZIG2 and ZIG3, and interestingly, gene set 1 genes were generally marked by HK27ME3, as I said earlier, and this was lost upon loss of both ZIG2 and ZIG3. On the other hand, when we look at ATAC-seq data, we observe that when we look at gene set 2 genes, there was a loss in accessibility in the vicinity of an ATAC-seq peak upon loss of ZIG2 and ZIG3, suggesting that these genes are regulated in very different manners by both ZIG2 and ZIG3. So to study this link between HK27ME3 and ZIG2 and ZIG3 potentially, because ZIG2 and ZIG3 bind a substantial proportion of these poised genes, and these are also de-repressed upon loss of ZIG2 and ZIG3, we conducted ChIP-seq of HK27ME3 in the single and combinatorial knockouts, and unsurprisingly, we saw very little change in HK27ME3 in the single knockouts. However, in the triple knockout, we saw a drastic change in HK27ME3. A lot of loci lost and gained HK27ME3. And a substantial portion of the loci that lost HK27ME3 overlapped with ZIG2 bound peaks, suggesting that there's a substantial dependence on ZIG2 for maintaining HK27ME3 with these loci. Then we further look into HK27ME3 peaks and stratified them into those that have ZIG2 and those that do not. What we observe is that a substantial portion, generally the loci that lose HK27ME3 are generally also co-occupied by ZIG2, whereas those that are not bound by ZIG2 generally don't have a loss in HK27ME3. Next, we wanted to get a sense of how convergent the roles of ZIG2 and ZIG3 and PRC2 are, at least with respect to the de-repressed genes. For this, we cross-analyzed our gene expression data with an EED knockout, which is a core subunit of the PRC2 complex. What we observed quite interestingly is that among the genes that are de-repressed upon loss of ZIG2 and ZIG3, a lot of these also are de-repressed in the EED knockout, suggesting that there's a convergence in function between ZIG2 and ZIG3 and PRC2, at least with respect to the genes that are repressed during prime pluripotency. Furthermore, to get back into sort of the transcriptome where we see an upregulation of genes that are overlap between genes that are upregulated in ZIG2 and ZIG3 knockout and the genes that are upregulated upon in vitro differentiation of the mesoderm and endoderm lineages. What we also observe is that a lot of the HK27ME3 bound loci that are found in primary HGSCs are also co-bound by ZIG2 and are lost upon in vitro differentiation into the endoderm or the mesoderm lineages, suggesting that this is this loss of HK27ME3 selectively over ZIG-bound HK27ME3 peaks is what's driving this transcriptome change to the mesoderm or the endoderm lineages. Next, we also observed that a lot of naive-specific genes were upregulated during this in our ZIG2-3 knockout, and we looked into transposable elements, and one of the transposable elements that was quite interesting to us were LTR5HS elements, and that is because these are hominoid-specific transposable elements that are important that are known to play a role in speciation towards the hominoids, and consistent with LTR5HS being upregulated, a lot of the genes that are known to be regulated positively by LTR5HS elements are also upregulated in the ZIG2-3 knockouts. Furthermore, when we look at what might be causing this upregulation of LTR5HS elements, we observed that KELF2 in particular was upregulated in the ZIG2-3 knockouts as well, and KELFs are known activators of LTR5HS transposable elements. And when we look into the HK27ME3 tracks of the ZIG2-3-5 knockout compared to the non-target control, you observe that there's a loss in HK27ME3 over these loci upon loss of both ZIG2 and ZIG3. Next, we wanted to delve a little bit deeper into the molecular mechanism. So for this, we conducted co-IP of ZIG2, and we found interaction with EZH2 with ZIG2. And we also conducted cut and tag of EZH2 in our wild type and ZIG2-3 double knockouts. And we observed loss of cut and tag signal of EZH2, especially over cluster 4 loci, which are again the ones that are bivalent promoters. But we also saw some loci over cluster 3 loci, although these have far lower levels of EZH2 to begin with at basal levels. And consistent with EZH2 playing a significant role in maintaining HK27ME3 via 6 in primed HSCs, we observed that when we look at these cluster 4 peaks, there's a strong correspondence between loci that lose HK27ME3 and loci that lose EZH2 occupancy in the ZIG2-3 knockout, as you can see by the scatter plot here. Next, we wanted to look at, again, what really got this process started, which is the accessibility of prime pluripotency and to what extent do ZIGs play a role in maintaining accessibility over these loci. So for this, we conducted ATAC-seq across all our mutant cell lines. And again, consistent with ZIG2 and ZIG3 having overlapping roles, we only see loss in accessibility over ZIG motif containing primed attack peaks in the ZIG2-3 double knockouts, but not in the single knockouts. And next, we also again conducted co-IP of BRG1 this time, and we observed interaction with BRG1 and ZIG2, suggesting that ZIG2 and BRG1 might play a role in keeping prime-specific attack peaks open in during prime pluripotency. So we mined published ChIP-seq data sets, and we observed BRG1 being very highly occupied over C3 loci. Again, these are the prime-specific enhancers that I referred to earlier. And we also see that these cluster 3 loci are also very dependent on BRG1 in maintaining accessibility. So upon loss of BRG1 in repriming conditions, so this is where you take naive HSCs and transition them to a prime state, they lose accessibility quite significantly upon loss of BRG1. Next, so consistent with BRG1 playing an essential role in maintaining accessibility over ZIG-mediated accessibility over prime-specific enhancers. When we look at ATAC-seq data, as you can see, cluster 3 loci generally lose accessibility upon loss on ZIG2 and ZIG3, and they also lose BRG1 occupancy over cluster 3 peak sets. This is not something that's seen across the other clusters, and is also further highlighted by this scatter plot here, where you can see most of these cluster 3 peaks being this bottom left quadrant. So essentially, if a given loci loses BRG1 occupancy, it also loses accessibility via ATAC-seq upon loss of ZIG2 and ZIG3. Next, we wanted to know to what extent do ZIG transcription factors play a role in exiting naive pluripotency. So far, my talk has been focused on prime pluripotency and sort of maintains our prime pluripotency. But now we wanted to know what happens if we topically express ZIG2 in naive HSCs which don't express any ZIG2 or ZIG3, and to what extent do they become more prime-like. So for this, we generated TED-on naive HESCs. So these are inducible naive HESC lines that can express ZIG2 upon induction with tetracycline. And we conducted ChIP-seq as a first pass on these cell lines. And what we observed quite interestingly and consistent with our endogenous ZIG2 chip in primed HSCs that ZIG2 mostly hones to prime-specific attack peaks, as you can see with the red line here, but they show very little enrichment of naive-specific attack peaks. So globally, we again did some cellular characterization, and what you can observe and hopefully appreciate is that upon ectopic expression of ZIG2, we observe this L-shaped transition from CD75 high, CD24 low cells, which are marker of naive HSCs, to a more prime-like CD24 high, CD75 low cells in a time-dependent manner. So suggesting that ZIGs do play a positive role in exiting naive pluripotency to more prime-like state. Furthermore, when we look at a global level at both the transcriptome and the chromatin accessibility landscape, we see a shift from a naive-like transcriptome and chromatin accessibility landscape to a more prime-like transcriptome and chromatin accessibility landscape. Next, to delve a bit more into the mechanism, we conducted again ATAC-seq in our DOX-induced TED-on ZIG2 naive HESCs, and what we observe is that when a given loci is bound by ZIGs, they become more accessible, and this is selectively found over prime-specific attack peaks that have ZIG motifs, but not over prime-specific attack peaks that do not have ZIGs, suggesting that this is a very specific, the phenotype that we saw earlier was mostly driven by a ZIG-dependent mechanism. And then when we conduct BRG1 cut and tag over these cell lines, over doxed new cell lines, we observe something very remarkable. So we see high enrichment of BRG1 over these flag ZIG2 peaks here, compared to vehicle, which don't have a lot of BRG1, suggesting that BRG1 is recruited de novo upon induction of ZIG2. And on the other hand, when we look at prime-specific, the loci that become open specifically upon induction with 6, they're occupied by BRG1 in the DOX-induced cell line, but they virtually have no BRG1 recruitment in the vehicle control cell line. Next, we wanted to get a sense of what drives these different behaviors of ZIG transcription factors across the 4 clusters. So for this, the first thing we did is we conducted a motif analysis of all the 4 clusters of ZIG-bound loci, and unsurprisingly, we found high enrichment of ZIG factors across all 4 loci, but the factors that they show co-enrichment for were quite diverse. So focusing just on clusters 3 and 4, we observed that cluster 3 have co-enrichment for SOX factors, which is another very strong activating transcription factor, and that ZIGs and SOX might interplay with each other to maintain accessible chromatin and active chromatin signature over prime-specific enhancers. Whereas cluster 4 showed enrichment particularly for E2F factors, which are found in PRC 1.6 complexes, and it's found generally over CPG-rich regions. And when we look at the CPG density, this can be further highlighted. So cluster 3 have very low CPG density, whereas C4 loci generally have a lot higher CPG densities. And given the inherent link between CG density and transcription activity, we observed that SER5P RNA pol 2 to total RNA pol 2 ratio was a lot higher over the C3 loci compared to C4 or any other loci. So to test this potential link between the transcription machinery and recruitment of ZIGs and ZIG-mediated activity, we treated our cell lines with triptolide, which is a small molecule inhibitor of transcription. And we found a very surprising finding in the sense that we didn't really observe any changes in recruitment of ZIG2 across all clusters 1, 2, and 4 upon treatment with triptolide. Upon acute treatment with triptolide, we observed far reduced recruitment of ZIG2 in the cluster 3 loci, suggesting that this is dependent on the transcriptional machinery selectively on the cluster 3 ZIG-bound peaks, but not the other clusters. And this can be further highlighted by these ChIP-seq tracks. So as you can see, this is the cluster 3 loci around the OCT4 locus, and there is virtually no ZIG2 signal upon treatment with triptolide. However, when you look at EVX1, which is found near the Hox cluster and has high enrichment for HK27M3, there's virtually no change in HK27M3. So I'd like to finalize, I'd like to summarize, summarize my findings from my work so far. And basically, what we observe is that ZIGs 2 and 3 are highly upregulated in transition from naive to prime pluripotency, and that they have very diverse roles across different chromatin contexts, and that over cluster 3, which are these prime-specific enhancers, ZIG2 recruits BRG1 and maintains these loci in an active open state, whereas in cluster 4 loci, which are bivalent promoters, generally they interact with EZH2, which is part of the PRC2 complex, to maintain HK27M3 over these loci. So I started off this talk with this Waddington's landscape model, and again, I hope what people appreciate is that we've identified some of the drivers of these drivers and barriers of these transition from naive to prime pluripotency, and then further from prime to mesoendoderm lineages. So ZIG recruits BRG1 to facilitate transition from naive state to a prime-like state, and also for self-renewal over prime-specific enhancers. And it also acts as an epigenetic barrier by recruiting PRC2 complexes and preventing improper commitment to the mesoderm and derm lineages. So finally, I'd like to thank our funding for obviously helping us funding our work. And I'd also like to thank my lab. Obviously, it's a team effort, especially my PI, William Pastor, and also the other people who've worked on this project over the years. I'd also like to thank our collaborators from the lab of Dr. Julie Lessard, who helped us generate a lot of the cut and tag data that was very important for nailing down the mechanism of some of these phenotypes. Thank you.
Thank you, Seth. Fantastic talk, very thorough, and I love the molecular part. While we wait for the question, which I'm sure will come in, I have a couple of curiosities about the interaction with PRC2, having worked on Polycom for a while before. So have you looked at the expression level, at the protein level of the PRC2 component and ZIC2 to begin with in the knockout of ZIC2, ZIC3?
Yes, we did look into this. The core components don't change much. The PRC 2.1 and 2.2 do change. So if I'm not wrong, EPOP, which is part of the PRC 2.1 complex, goes up, whereas JARET2, which is part of the PRC 2.2 complexes, go down. But the core subunits don't change much at all, at least at the level of friendship.
And slightly following up on that, have you tried to see whether the interaction with PRC2 and EZH2 is direct, or if there's any mediator in between?
So we've been trying to come up with a few experiments that looks into this a bit deeper. We haven't done the experiment yet. We're trying to work with PLAs and like short arms and long arms, but it's, you know, we just haven't come across the right protocol to really properly test that yet.
Okay. Yeah, no, it's a tricky one, but it would be very interesting mechanistically to see whether where there is any factor on the. And well, then I'll take another one since I have space. Regarding the motifs of the mediating the binding to DNA. Have you found anything that resemble other classical Polycom recruiters in your I don't know if by cluster or overall, or this kind of if there is anything else that you find? So it seems to be acting as a Polycom recruiter, right?
Yeah.
So have you find anything else that might look like it in your picks?
In terms of the motif analysis, this is what we have really. Besides that, I'm not aware of, not really, at least not within the Homer motif database. This is all we have. And we generally know that basically polycomb bound loci generally have a higher CpG density, as you can see here.
Yeah.
But beyond that, in terms of a specific factor, we're not really sure. We haven't really found a particular motif that corresponds to a factor, if that's what you're asking.
And then I'll take another one. I seems to have seen at some point in one of your K27 tracks when you had the Zik mutant, that the reduction seems to be on not at the center of the peak, or more on the flanking parts of the peak rather than the center. Exactly. Okay.
This one.
Yes.
Okay.
So no, okay, no, it's minimal. Maybe it wasn't this one. Okay, yeah, never mind. It was an impression that I had that there was a differential effect of the K27 trimethylation reduction on the edges or the flanking side of the K27 trimethyl peak rather than the center. I don't know if you notice anything like that.
So generally we observe only around the centered on the ZIC2 peak. So we see a reduction, say if a polycomb HK27M3 and a ZIC2 peak, the ZIC2 peak is very sharp, whereas or relatively sharp compared to the polycomb loci. So we see a very punctate drop, definitely a very intense drop for most cases of HK27M3 over that punctate loci around the vicinity of the ZIC2 peak, whereas the flanking loci generally have a lesser reduction, I would say. However, we do see certain loci where there's complete loss of HK27M3. Now, whether that's driven only by ZIC2 or whether that's driven by, say, another transcription factor coming in, which is particular to a certain lineage, activating it and removing it further, I'm not sure. But we mostly see cases where there's definitely a dip, a more intense dip around the vicinity of the ZIC2 loci and less so towards the flanking loci. But we do also see cases where it's completely lost.
Okay, fantastic. We do actually have a couple of questions that came in. So Jason Fan was asking, I wonder if BRG1 occupancy is also altered in the triptolide treatment for cluster 3, and whether you could speculate the mechanism of how ZIC2-3 binding is lost with triptolide inhibition of transcription.
Yeah, this is a very interesting question. We really like this experiment in the lab, but we're really delving into the mechanism a bit more. I'll just get back here to kind of, yeah. So we didn't do that specific BRG1 experiment, although it would be very interesting. I think there's a preprint by Steve Henikoff's lab where they do show dependence of BRG1 on nascent transcription. So they did experiments with triplet and flavopyridol. We also tried doing similar mechanisms where we tried to narrow down basically aspects of transcription. So not only just transcription in general, which is what triplet does, but the RNA, the methylated RNA, and then read through of RNA pol2. There's a very sad so what happened was we actually also did those experiments, or we were trying to do those experiments, but then our sonicator kind of broke down, so we didn't really have good data from that. But we are basically, if I were to do the experiments again, I'd look at not only just transcription, but transcriptional read-through. So, use something like flavopyridol or alpha-amanitin, or I think actinomycin. And then I'd also look at just the RNA itself, whether the RNA itself can play a role in recruiting transcription factors, because I know there's another preprint by Rick Young's lab, which gets into that a little bit. So I have we have a few theories, and we did try testing them, but unfortunately our sonicator broke down, but we'll definitely do it when the opportunity arises very soon.
Nice, thanks. So
Thank you.
the next question is from Nicola Maresca. I wonder what happens to the cells in the knockout for Zik. Do they differentiate into different lineages?
Yes, yes,
do the Zik work as a pioneering factors?
Yes, so I'll answer the first question first. So when we look at the transcriptome, they mostly give rise to the mesoderm and endoderm lineages, and this can also be seen by our flow cytometry data here, by CD50 CD184 staining. We do see some change, some increase of the naive-specific genes, which might be driven by things like LTR5HS, for example. So they definitely lose their primed identity, and if I were to say, they become probably more mesendoderm-like. But they also have certain genes that are expressed highly in naive HCCs as well. So we see SMARCA2. Yes, but that's what I would say. The mesoderm lineage is dominated with some naive HCC signatures. And the other question in terms of pioneering factor, the thing is, we did this. The best system to really study this would be the TED-ON system, which we already have. But the issue is we never really did. We would have to do more experiments to really say that, I feel. I'm sorry, I'm on the wrong slide. For example, time course experiments and the like. For example, if I were to do that experiment, I'd do maybe a couple of histone marks and factors like BRD4 and BRG1, but I don't think we have enough data to really say whether it acts as a pioneering factor just yet. It potentially might, and it looks like it might, but I'd have to do more experiments before I can really conclusively say that.
Fantastic, thanks a lot. So we have to move on due to time, but if you want, there are a couple of other questions that you might type and answer to in the chat box, in the Q&A box, sorry.
Awesome, thank you so much for your time.
Thank you very much for this fantastic story.